Okay, good morning, students. I hope that the screen I've shared is visible to you all.
Yes, ma'am.
Okay, so today it's going to be the last class in this course, and Friday we can have some amount of discussion, or if there is something that you would like to ask, we can go ahead on Friday. So today what we are going to look at is a different class of molecular machines. So far we have seen molecular machines as they were found in nature. For example, we've seen translocation motors, we've seen polymerizing motors, we have seen rotary motors, we have seen motors that can unentangle double-stranded DNA. But today we are going to look at a class of molecular machines which have been artificially designed. So let's go ahead with that. So, the molecular machines that I'm going to talk about today, all of them are made out of DNA. And DNA is very versatile in the sense that you can exactly create different structures if you can modulate the sequence. Because we know that DNA can form very specific base pairings, and therefore this property can be used um, to make very specific base pairings and very specific sequences or sequence secondary structures. On top of that, you can make these DNA structures of various lengths. You can make them short oligonucleotides, you can make them really long. So, in that sense, they're very versatile. And the size of these DNA double stranded structures is such that they can be used as probes in silico analysis, they can be used as probes even inside the cell. So, we're going to look at two such examples today. So to begin with, this is a classical diagram that explains to you the Watson and Crick DNA double helical structure. And this is just to recap. So you have the two DNA strands running anti-parallel. And if you think of this double helical DNA as a cylinder, then the diameter of the cylinder would be 2 nanometers. And as the double helical DNA, for every one complete turn of the double helical DNA, you will have approximately 10 of these base pairs. And so this complete turn is about 3.4 nanometers. So why I'm saying this is because we are going to come to this in the next few slides. So this 3.4 nanometers, please remember this. So one turn is about 3.4 nanometers. So 1.5 turn would be a little less than 5 nanometers. So there is a kind of periodicity to this DNA structure, and that can be useful to design some machines. Okay, so the machine that this group has tried to design is based upon the walk of the kinesin motor. So, what are the properties of kinesin motors that you can remember? Anything that you remember about kinesins? Ratnesh?
Yes, ma'am. Like it was a step by step, and there is like its head, and then there is a structure. It have. Uh,
Okay, so it has a hand-on-hand kind of movement. So is the movement very periodic in the sense that are the step sizes very uniform or the step sizes are unequally distributed? So it can take a very large
Ma'am,
step,
in case
small
of
step.
A kinesin, we have a step sizes like equal. In case of dynein, we don't had equal
Right.
steps, and I remember as well.
Absolutely right. So for kinesins, if you remember the structure, and if you think of dimeric kinesin, so there can be monomeric, dimeric, tetrameric kinesin. So if you think of a dimeric kinesin, you had a helical structure that helped it to form this dimer. And then at the end of the dimer were the 2 feet that were stepping on tubulin and polymerized tubulin, so or microtubules. So and every time it walked, it had a very periodic movement. That is, the step sizes were very uniform, and that depended on the periodicity of the alpha beta tubulin dimers on this microtubule. And so, other than the cargo carrying portion, you can think of it very simply as having this double helical region, the coil that helped the kinesin to form homodimers, and then you had the 2 feet to walk. So that's essentially the minimum unit that is required to walk on the microtubule. So these authors, they tried to mimic that and mimic the walking of a kinesin on microtubule by simply using DNA. So they designed DNA in such a way that the DNA itself will form the microtubule track. The DNA itself, another piece of DNA would form the kinesin-like structure, and then you have to somehow be able to create the periodic movement. And then you need to be able to visualize that periodic movement, because how will you be sure that what you have designed or what you hypothesize is actually working? So you need a way to be able to observe this movement. So this is something very similar to a kinesin. If you think of kinesin, you have a coil domain that helps a dimeric kinesin to form homodimers. So you have these coils. So you have 2 strands of DNA with the ends forming these double helical regions, and the ends that are complementary to each other will be about, say, 20 base pairs. So that can form this double helical structure. 20 base pairs would be approximately 2 helical turns, complete helical turns. Okay, and after that, you have about 23 base pairs of region on either strand, which will not be complementary. Therefore, it can form this kind of a structure, and it's very analogous to kinesin. You have this double helical structure, and then you have two other domains that bind to the microtubules. Here you have these 2 single-stranded regions that would be capable of forming base pairs with another piece of DNA. Now, in order to facilitate our observation of the walk, what they've done is they've added 2 fluorescent quenchers, 2 different fluorescent quenchers at the ends of this single-stranded piece of DNA, these ends. So you have one fluorescent quencher here, another fluorescent quencher here. Now, what is a fluorescent quencher, if anybody may know? So what is quenching?
We quench the fluorescence part, like fluorophore is attached with the quencher, and when the quencher is removed, we can get a fluorescence. So these
Right.
quenchers are some.
So a quencher would be something, if I say a fluorescence quencher, it would be something that would have a very big, either very big overlap with the emission spectrum of the fluorescent molecules, such that it will be able to absorb all the fluorescence and you will not be able to see a detectable fluorescence. So again, it depends on the electronic structure. You could have different types of quenching. You could have quenching not because of a molecule, but because, say, of oxygen. So if it is oxidized, the entire structure of your fluorophore might change. But here, the quenching that is used is based upon close proximity. It's kind of analogous to FRET, if you think of it. But in FRET, you had the acceptor molecule emitting a second fluorescence. Here, if you have to think of the quencher as being able to absorb the emission, but it does not quite fluoresce itself. And again, this is also dependent upon the close proximity of the donor of the fluorescence of the fluorescent chromophore and the quencher. So the closer they are, the more the quenching effect would be seen. They added two quencher molecules here. So if they've added two quencher molecules here, the fluorescence must be added somewhere else. So this is then the microtubule-like track that they have designed, again using DNA. So this piece of microtubule-like track has been designed by using 6 different oligonucleotides. 6 different oligonucleotides such that, so just observe this one. So you have this one green strand that partly forms a double helical structure, and then the rest of it forms a single stranded piece of DNA. Again, you find this, this another purple, it forms a partly double helical structure, and then again a single stranded piece of DNA. Similarly, the blue one, another, say, violet or blue. And then you have 2 other green and light violet structures that do not have these single-stranded piece of DNA, but rather help in creating this double-stranded scaffold. So 4 of these, out of these 6 oligonucleotides, 4 are essentially used to make these single-stranded regions of DNA, but in such a way that the principle is again the same. You have a part of it that forms double helical structure and the rest of it remains single-stranded. And at the end or tip of each of these single-stranded pieces of DNA, you have a fluorescent molecule. So the idea is this: this Walker, by some means, is able to hybridize. What is hybridization? It's able to form double-stranded DNA with this strand and this strand. Then this will bring the quencher molecule in close proximity to the fluorophore, and you will no longer see the fluorescence. So what you're observing here is the reduction of fluorescence every time the Walker single-stranded parts of the Walker hybridize to the single-stranded parts of the track. Why? Because you have added quenchers to the end of the single-stranded pieces of DNA and you've added fluorophores to these ends of DNA. So far, is it okay?
Yes, ma'am.
Yes, ma'am.
So itself could end up hybridizing our Walker to the track, but it's not. If you would do that, then how would you be able to remove the Walker? Because it's a Walker, so it needs to walk on the track. Then how would you be able to remove this? If you were to simply make a base pairing of this with this, a direct base pairing, it would be difficult for you to remove it. So what they've done is they have used 2 different additional oligonucleotides. And here is the trick. So they have used one oligonucleotide, they've named it A1. So this single-stranded piece of DNA partly hybridizes with the Walker, partly hybridizes to the track. And between the Walker and the track, these 2 regions, there is a little bit of piece of single-stranded DNA. Plus, you see that it does not completely hybridize to the Walker. There is a piece of DNA even at the tip of the Walker that remains single-stranded. And this is essential because this allows the Walker to have some amount of flexibility. Flexibility is important if it needs to walk on this track that has been created with the help of DNA. So all these hinge regions, if you remember, even in the case of myosins, dyneins, kinesins, you had a certain hinge region that gave it a flexibility. So this hinge region is created here by the means of having some a certain stretch of DNA that does not form double-stranded region with this additional single-stranded piece of DNA. So this they have termed, is a term that they have used as attachment fuel strand. So this is an attachment strand, or A. And the idea is this. So you have added the Walker. Suppose you have taken a flow cell and you add the track, this track of DNA, and you add the walker, because these two strands do not really complement each other, the walker would not bind on the track. So you would always see very high fluorescence of all of these fluorophores. You'll always see high fluorescence. But the moment you add the A1, it would hybridize partly to the walker, hybridize partly to the track. This would bring the quencher closer to this fluorophore, and there would be a sharp decline in the fluorescence of this particular molecule. You would still see fluorescence here, fluorescence here, fluorescence here. So only this fluorescence would be specifically quenched. Now, similarly, you could add another short single-stranded oligonucleotide that hybridizes partly to this strand of the walker and this strand of the track, while leaving a little bit of single-stranded region here, that's the hinge for mobility of this walker, and that would again hybridize this strand to this, and you would see decrease in fluorescence of this fluorophore because this quencher molecule is now close to the fluorophore. So that would give you a situation like this. This is the second situation that you have. Now, so far we have seen how you can make the walker hybridize on this DNA strand. But then how does it walk? Because the name is walker, it should be able to walk. So in comes something called it's their terms, they have called it the detachment strand. So there is an attachment strand and a detachment strand. And what the detachment strand does is, it is designed to be a complement of the attachment strand that was used. So whatever you have used here, this strand, you make an exact complement of this strand. And so you're expecting a strand displacement kind of reaction. If there is a strand displacement kind of reaction, because there is an exact complement and it would like to hybridize with this strand, then the strand would be released and the walker would also be released off the track. So this walker was initially linked to the track because of the attachment strand. Attachment strand did not have complete complementarity to the walker. It had regions which formed single-stranded DNA because you needed a part of it not to complement with the walker to give the walker some amount of flexibility. You also had parts here that did not complement. Now you add something that would completely complement with this, and therefore this can cause a strand displacement reaction, and of course this can be enhanced with the help of temperature. You enhance the temperature and then this would be released and would rather bind completely to a complete complementary piece of DNA, and so this part of the walker would be released. What would that do? That would leave your walker hinging on just one piece of one of its leg. So instead of being bound to instead of being bound by both the legs, now it is bound by one leg. And now if you add a second attachment strand that rather complements with this, then you can have the entire molecule move from this step to this step and so on. So basically you have created ways by which it can hybridize, release, and every time it hybridizes and releases, it can move one step along this piece of DNA. And you can monitor every time it hybridizes by looking at the fluorescence quenching. So as long as these strands are free, you will see fluorescence. When the strand hybridizes, you will see quenching, you will see absence of fluorescence. So in this condition, both of these fluorescence are absent, and you will see fluorescence only from these two fluorophores. In this condition, this fluorescence is regained because the attachment has been lost. But this quenching persists, so you will not see fluorescence from this, or you see very low fluorescence, and you will now see fluorescence from 3 signals instead. Or is it okay? Or should I repeat this once more?
Ma'am, can you please repeat this just
Okay.
briefly?
Okay. So let me introduce this once more. So the entire game here, if I say it in Hindi, that पूरा पूरा जो चीज है वो इसके ऊपर डिपेंड करता है कि आप डीएनए के कॉम्प्लीमेंट्री स्ट्रैंड्स ढूंढ रहे हैं. So DNA would like to form these complementary base pairings. और इसी को लेके ये पूरा डिजाइन किया गया है. पहला डिजाइन क्या है? पहला डिजाइन वॉकर का है. आप डबल स्ट्रैंडेड डीएनए ले रहे हैं, लेकिन डबल स्ट्रैंडेड डीएनए इस तरह से है कि एक साइड तो डबल स्ट्रैंडेड बन गया, लेकिन दूसरा साइड सिंगल स्ट्रैंडेड रह जाता है. So these two strands are now free to form double helical pieces of DNA if they find complementary strands somewhere else. और इन दो strands के ends में क्या लगाया गया है? फ्लोरेसेंस क्वेंचर. ये वॉकर है. यहां तक ठीक है?
Yes, ma'am.
तो अभी ये ट्रैक है. ट्रैक कैसे बनाया गया है? ट्रैक का बहुत सारे डिजाइन कर सकते हैं, बहुत सारे ऑलिगोन्यूक्लियोटाइड्स डाल सकते हैं. मिनिमम आपको क्या चाहिए? आपको ये मिनिमम यूनिट टू शो वॉकिंग है. सो मिनिमम यूनिट में क्या दिखा रहे हैं? यू हैव जस्ट लाइक दिस, यू हैव क्रिएटेड regions of double-stranded DNA. Then it is followed by regions of single-stranded DNA. Again, you have a region of double-stranded DNA, but then there is a single-stranded DNA. So essentially, all of these lines that you see in color shows one strand. The other strand is another piece of DNA. So ऐसे करके करके आप इस तरह से double-stranded regions में specifically single-stranded pieces of DNA निकाल सकते हैं. So it's about having multiple DNA strands that can form double helical structure, but at the end it is single-stranded because it doesn't have a complement. Now you bring another piece of DNA that forms a double helical structure, then another stretch that is single-stranded, so it doesn't have a complementary strand. You bring another strand. ऐसे करके करके you can create long stretches of DNA, double helical followed by single-stranded pieces of DNA. Again, double helical followed by single-stranded pieces of DNA. Essentially, what it would look like is like a long cylinder with very uniformly spaced regions of single-stranded DNA. Now, this is important. I had missed this. So they designed it in such a way that each of these single-stranded pieces of DNA occurred exactly after 1.5 helical turns. After 1.5 helical turns, so that all of these single-stranded pieces of DNA face the same side. ऐसा नहीं है कि one is facing this side, the other is facing this side. So all of the single-stranded pieces of DNA, you can think of it as being like a long cylinder, and you have single-stranded pieces of DNA emanating out of that cylinder. And at the tip of each of these 4 strands, you have 4 different fluorophores. So you can individually monitor the fluorescence of each of these strands. Now, if your Walker can somehow hybridize on the strand, then this quencher will come very close to the fluorophore, and the fluorophore would be quenched. That is, you will no longer see the fluorescence. So if this quencher is close to the fluorophore, you will not see the fluorescence from the fluorophore. So ये डालने से पहले the fluorescence signal was high for the green. After you have hybridized this, the fluorescence signal for green will go down. But this strand doesn't form a direct complementary strand with this. Rather, you have a second piece of DNA, single-stranded DNA, that partly hybridizes to this end, partly hybridizes to this end, and can bring the Walker in close proximity to the strand. So the other important thing is that it will have regions which are single-stranded. So this region is still single-stranded, this region is still single-stranded. So not the entire piece of the third or the attachment strand is completely complementary to both of these. There would be regions which are still single-stranded, and that is useful if you want to detach this attachment strand. So now if you add a strand that is completely complementary to this strand, you increase the temperature, you release this. Now this, instead of hybridizing back to this, it has found something which is completely complementary to it, and it would like to go and hybridize quickly to that, leaving your walker end free. So once this is released, again the walker will be free. So again you will see the fluorescence of this signal because the quencher will no longer be close to it. Think of this situation. You had the walker molecule hybridized to both of these strands using two different oligonucleotides. Two different, because these two sequences are different, these two sequences are different. So you designed another two pieces of oligonucleotide such that the two strands of the walker can hybridize on these single-stranded tracks. Now, if you add the detachment strand that is completely complementary to this attachment strand, and you are releasing this by strand displacement, then this leg is now free. Once this leg is free, if you add a second attachment strand that will hybridize to this, but instead of this it will hybridize to this, then you can make the entire walker move from this position to this position. So it will now bind to these two. So it has moved one unit in front. So by just adding the attachment strand, then the detachment strand, then the next attachment strand, then the next attachment strand, you can make this walker move at regular spacing on this track. So far, is it clear?
Oh, yes, ma'am.
So this gel essentially shows you whether they have been able to, so this is hypothesis. This is all, say, designed on paper, on computer, just by looking at the sequences, trying to see if they would form double helical structures, trying to see the probability of them forming very tight helices and so on. But when they designed this, and next thing is to test it. So the first thing they did was they took the track and they tried to see if the track itself, that is this track, they have hybridized all the oligonucleotides. They expect a structure like this. And they expect that the walker will not hybridize onto the track because there is no region of complementarity. So they tried to test this simply by running a gel. So if they run it on the gel, if you have the track molecule running here and you add walker, if the walker had hybridized onto the track, then the entire molecular mass of this track plus walker would be higher than the track, and so you would have a band much higher than this. Instead, they'd see that the two bands are at the same position, which indicates that just by adding the walker on the track, just if you add this walker and the track, the two do not hybridize, the two remain separate. Why are you seeing just one band here and not two bands? You have added the walker. You should have been able to see the second band that is formed by the walker. You do not see it because you're specifically detecting the fluorescence associated with the track. Because remember, we have added fluorophores to the track, quenchers to the walker. So you're observing this hex detection is basically you're detecting one of the fluorophores. So when you add the walker, the walker does not hybridize onto track until you add the attachment strands. So you add the attachment strands and you see that the molecule now forms a higher molecular mass structure. If you add the detachment strand now, so you've added A1, added A2, you add the detachment strand, you see that the molecular mass of this entire complex goes down. You add another attachment strand, it goes up. So this gel is showing them that at least the hybridization properties are going according to their plan. But that does not prove that the molecule is indeed walking. So for that, they used a method to detect fluorescence in real time. So what did they do? They first took this track. Now the track has 4 fluorophores, right? We will call them A1, A2, A3, A4. after the 4 attachment strands that can be bound to each of these. So just for simplicity, we'll call this fluorophore A1, A2, A3, A4. And for each of these attachment strands to get detached, you need the detachment strands, and we'll call them D1, D2, D3, D4. So correspondingly, detachment strands. So initially, you can think of a situation where you have added the track, added the walker, and added A1, so that it just hybridizes to one of the strands. This is the initial setting. If you want to do this initial setting, that is this is the point from which you will start observing fluorescence, then what do you expect? You expect to see very high fluorescence of A2, A3, A4, but not of A1, because A1 is getting quenched. Is it okay? You've added the walker, you've added a track. Just the walker and a track will not hybridize. You should see all 4 signals. But if you add this first attachment strand, then the quencher and fluorophore would be together, in close proximity. You shouldn't see high fluorescence from A1, but you will see high fluorescence of A2, A3, and A4.
Yes.
This is the first initial position here. This green is for branch 1, that is A1. And you see, this is low. All of the others, A2, A3, A4, all of them are very high. This is the initial start point. Now they add A2, the other strand to it. So all of this requires controlled fluidics. This is kind of a controlled fluidics experiment combined with real-time fluorescence detection. So now if you add A2 to this mixture, what do you expect? You expect that the fluorescence of A3 and A4 will remain unchanged. Because A1 is already attached, A1 fluorescence would still remain low, and A2 fluorescence would go down drastically, because now the A2 has hybridized. So the situation would be this. So A1 was attached, this fluorescence was quenched, you had these 3 fluorophores. Now you've added A2, so A1, A2 both will be quenched, but A3, A4 would remain. Exactly, that's what you see here. So 2 fluorophores, A3, A4 are still fluorescing, but the moment you add A2, A2 fluorescence goes down, A1 was already low. Now, in this condition, if you are able to release this attachment strand by adding the detachment strand, if you remove this attachment strand, then this fluorescence would again come up, will be high, because the fluorophore is no longer close to the quencher. You'd have high fluorescence of this fluorophore. This fluorophore is still quenched, so A2 is still quenched. A3 and A4 will still show high fluorescence. So if you see this analogy here, you add A2, the attachment strand, both the legs are now attached. So A2, A1 fluorescence is low. The moment you add the first detachment strand, the A1 is now free. So the A1 fluorescence goes high. A2 is low, and A3, A4 remains high as usual. So that is the case here. You have this case where A1 is now free. But now if you add another attachment strand or another molecule, that oligonucleotide, that will hybridize to this and hybridize to this, that is the A3, then you should see quenching of the third fluorophore, but the fluorescence from 1 and 4 should be high. 2 is anyway quenched because it's still attached to the molecule. So that's here. You add A3 and you see very specifically the third fluorophore fluorescence has gone down. A2 was anyway low. A1 had been released, so A1 is high. If you add D2, now the second leg should be released. If the second leg is released, what do you see? You see that after addition of the second leg, A2 fluorescence that was high is, if you add D2, one second, let me trace the branch. Why is this? Have I confused it? This is branch 4. Branch 4 fluorescence goes down. Okay, D2 is where the fluorescence should be up. Sorry. So it's all right. So once you have added the second attachment strand, the fluorescence of the second D2, A2 should go up. So that's exactly what you see here. So in this manner, you should be able to clearly visualize the attachment and the detachment. So every time it attaches, every time there is attachment, you see decrease in fluorescence. Every time there is a detachment, you see increase in fluorescence. So in this manner, you are able to observe the specific walking movement of this molecule on the track. Now I'll keep it on the slide to see if you have any questions. Any questions?
Ma'am,
Yes.
when they increase the temperature, how do they make sure that only one strand is detaching from the two of the hybridized strand?
Well, this is the whole key thing here. So the entire program that they have used to design these sequences would determine what would be the melting temperatures of each of these double-stranded pieces of DNA, and has to be designed in such a way that if they are using strand displacement, then definitely the TM of these strands should be lower than the TM of the strands they do not want to dissociate. So
Yes.
the first part of this entire the idea is novel, the idea is unique, but the whole idea, the whole work here comes up where you have to design these sequences. It's not a simple task to do it, and they have used programming to do it because it's not possible to do it manually. So they have used certain softwares to understand how they can specifically change the TM of one, keep the TM of the two pieces of strands quite far apart so that they're able to specifically dissociate one strand while keeping the other strands complementary. And when you dissociate it and you try to reassociate, this strand would not reassociate because you're giving something else that is a perfect complementary to this. So it would rather bind there than to bind back to the walker. So
Yes.
the whole idea hinges on very perfect designing of the sequences they have used for each of these constructs. That is the key in this entire experiment.
Yes, ma'am.
Okay, are there any other questions? Is this part clear to all? How they have observed the walking by just tracing which fluorophore is getting quenched and which fluorescence is actually revived or is visible once more. So every time the fluorescence is getting quenched, the strands are hybridized to the track. And when the strand is released, that is when you would see the fluorescence being regained. If there are no questions, we'll go into another interesting piece of. So this is the paper from which I have just taken this beautiful work, and we'll now move on to another way by which you could use DNA as some kind of a molecular machine that would give you some kind of readout. So first was where you were observing DNA being structured in such a way that it is able to mimic the walking of kinesin. So the walking of kinesin is unidirectional. So you can make this unidirectional. How? By specifically adding one. So you can make this motor go in the reverse direction if after adding A1 and A2, instead of adding A3, you add back A1. So it would just step back. But you can make it walk like kinesin, walk in one particular direction by sequentially adding the attachment strand 1, then 2, then 3, then 4. You ensure that it's going in one direction. So just like kinesin. Again, just like kinesin, it has very regular spacing, and the regular spacing is obtained by the regular spacing of the single-stranded regions on this DNA track. So if you make them unequally spaced, you can make them walk instead like a dynein. That's also possible. But they have tried to mimic kinesin walk by making this spacing very uniform. So after every 1.5 helical turn, there is one single-stranded stretch of DNA to which it can hybridize. About DNA as a walker. Now here DNA is used in such a way that it can detect local pH changes. Now this is a fantastic tool because very often we would like to understand what are the pH changes happening inside the cell. For example, you might know certain regions of the cell that have very distinctly low pH. Can you name any region that might have a low pH inside the cell?
Lysosome.
Lysosome, right. So lysosomes are organelles which have mechanisms to continuously pump in protons that ensures that the internal pH of the lysosomes is always lower, as low as 5.5. So that is much lower compared to the cytoplasm, which is nearly neutral. So if you think of understanding what are the subcellular pH of a region, or even in the cytoplasm, if you would like to see which regions have a distinctly different pH, you use certain dyes. And how do the dyes respond to pH changes? They respond to pH changes by either getting protonated or deprotonated. So if it is at a very low pH, it would be always protonated. As the pH goes higher, the pH indicators would start getting deprotonated. So between protonated state and deprotonated state, you could have a difference of color. You could have a difference of, if they are fluorophores, you could have a difference of either fluorescence emission spectra. Sometimes the emission spectra might differ. Sometimes emission intensities might differ. So you could have all of these changes guided just by protonation, deprotonation events. Now, what if you would like to understand, suppose there is a protein and the protein is being internalized into the cell, and you would like to know where in the cell does it go? How does it go into the cell, whether it is degraded, whether it is pushed back outside the cell? Many a times, proteins that are on the surface of the cell, like certain receptor proteins, when they are activated by the ligands, they get internalized. Why should they be internalized? Because the receptors are acting like antenna. They are there to capture the signal. Once the signal is caught by them, what they would do is relay the signal inside the cell. That is called signal transduction. They would relay the signal inside the cell. So there would be additional proteins, adapter proteins, etc. coming together, and you would have these signals being ultimately transmitted to the nucleus to either cause some transcriptional regulation. Say some genes are expressed or some genes are repressed. But if you have an antenna like this on the surface of the cell, and the antenna, once it has received a signal, is always on, then that is harmful for the cell. You cannot have any signal always on. So for example, your cell has detected that in the environment it has a lot of insulin. So once it has bound to insulin, if it keeps the insulin receptor signaling always on, then that's bad, because there might be a situation where insulin is low and it has to respond in such a manner. So one of the classical mechanisms by which the cell dampens this signal or blocks the signal is by internalizing the receptor and then either destroying the receptor or getting rid of the ligand, that is getting rid of the signal from the receptor and pushing just the receptor back to the surface. So both of these could work. Now, for you to understand whether there is an internalization, whether it's getting degraded, whether it's going back to the surface, you would want to observe these receptors, these proteins as they are going inside the cell. And very often these internalization events occur in these vesicles, and the vesicle pH can often change. Suppose the vesicles get fused to lysosomes, then you will see the pH of the environment in which the protein is now present is very low. So various reasons for which you would like to see local pH changes inside the cell. And there are other ways to do it, but this is one of the fantastic methods by which you can observe such pH changes. So what is the construct that they have used here? Again, it's a piece of DNA, but you see it's made up of 3 oligonucleotide strands. So single-stranded pieces of DNA, 3 strands. You have strand 1, strand 2, this is O2, and the strand 3. And you can see from the picture that part of the strand 1 hybridizes to strand 3, part of the strand 2 hybridizes to strand 3. So strand 3 is hybridized to both 1 and 2 such that you have 1 or 2 base pairs that remain single-stranded in between. So you have this little single-stranded region of DNA. And as we have seen in the previous experiment, that little single-stranded piece of DNA provides a little bit of flexibility to this entire molecule, such that it can deform and it can bend and buckle like this. So if it bends and buckles like this, then if you add 2 fluorophores at the 2 ends, these 2 fluorophores can come together. In this structure, they are very far apart. But if this entire structure bends, then these 2 fluorophores would come together. And once they come together, if you choose a Fret pair, so if you choose a donor-acceptor pair, then at this condition you will see 0 Fret or very low Fret. In this condition you will see very high Fret. So they have used this construct and they have designed it in such a way that if there is low pH, if this construct is protonated, then it tries to assume a shape like this. It tries to bend and assume a shape like this, because then these single-stranded pieces of DNA can form some base pairings, non-Watson Crick base pairings, and can emit Fret fluorescence. That is, the emission from the acceptor molecule can be seen. So this is the entire construct. High pH, low pH. High pH, you will have low Fret or no Fret. Low pH, you will see Fret. So if you now just observe, so what is shown here in these curves is basically the Fret ratio. So I have explained previously in both the previous lectures that when we see Fret, what we do is we do a ratio of the emission of the acceptor versus the emission of the donor. If there is Fret, then the emission of the acceptor will be very high and the emission from the donor will go down. So this ratio will be very high. But if there is no Fret, then the emission of the acceptor will be very low, emission of the donor will be high. So the ratio between acceptor fluorescence and donor fluorescence will be very low. So that gives you a Fret ratio. So again, what they did is they plotted this ratio over different wavelengths, because there is a characteristic wavelength at which these fluorescences can be seen, and they found that this is just looking at what should I say? This is just looking at the fluorescence of the acceptor molecule. And you see the fluorescence of the acceptor molecule goes up, the intensity goes up as you go down the pH. So this is between pH 4 to pH 7.3. So here is pH 4, here is pH 7.3. So you see very high fluorescence of the acceptor at low pH. And if you do a ratio between donor or acceptor, you would see that the fluorescence energy transfer is maximum at low pH, and minimum at pH that is around the cytoplasmic pH, nearly neutral. So this is one example from the paper. The paper has used multiple ways to actually show that you can really use this construct to observe pH changes in the cell, in a living cell. So while the cell is moving, dividing, over days, you can watch the fluorescence, changes in fluorescence, internal changes in fluorescence that shows internal changes in pH. So this is one such example. So they have designed it in such a way that this entire construct, can be endocytosed by these cells. Do you know the meaning of endocytosis? Endocytosis, anybody? Have you heard the term?
Ma'am, it is something like mitochondria and chloroplasts are where bacteria and they get inside the eukaryotic cell. I don't remember correctly.
Okay. Yes, it is an evolutionary theory that mitochondria and chloroplasts, because of their similarity in structure and the similarity in their DNA and in some of the proteins that are present, that they could be ancient bacteria that got engulfed into eukaryotic cells. But the idea, you're right. But what is endocytosis? Endocytosis is a process by which cells can take in from the external media certain proteins, certain ligands, certain some external components into the cell. So there are specific methods by which endocytosis could take place. It could be clathrin dependent. So there are some pit-like structures. So what is required for endocytosis to take place? Either the cell needs to have cup-like structures on the membrane, and then if there are these cup-like structures, these cups can close and get pinched off from the membrane, and the entire vesicle can move into the cell. So those cups are stabilized by certain cytoskeleton proteins called clathrin. The whole idea is that it's a method by which you can engulf. You can have, so there are different ways you could do this. You could do it by pinocytosis, endocytosis. So at the end, it's a very controlled process by which essentially you have the cell surface forming cup-like structures, and then this cup gets pinched off from the membrane and it's internalized. So anything that is at this cup, say there are receptors at this cup, or anything that is bound to the receptors at this cup, or if there is liquid medium here, then all of that can be pinched off and get into the cell. So this is endocytosis. So they use this construct and followed it as it was endocytosed, as it was taken up by these cells, and they followed it as it moved into the cell. And once it moves into the cell, it can move into certain structures that can have very low pH or can have neutral pH. So once the proteins are endocytosed, it may not immediately go into a structure that has very low pH. Or sometimes it may persist in structures that have very low pH. So what is happening to this? So how is this being trafficked, or how is this being, is it immediately going into low pH structures? Does it get degraded in the low pH structures? Does it remain stable in low pH structures? So here what they have observed is that once the endocytosis takes place, this molecule gets kind of stored in certain structures called late endosomes. And they found that the pH of these late endosomes can be quite low. That is what you see here. So this is just a pseudo coloring to show you the fret intensity. So very high fret at low pH, low fret or zero fret would be seen at pH which is near the cytoplasmic pH. So you see in the cell, wherever there is almost, all of these dots, whatever you see here is this molecule emitting fret. And you see that in some cases it shows very high fret, some other cases it shows very low fret. And the cases where it shows very high fret, would be regions where the cell has a very low pH. And to do this, they added this piece of DNA into the cell, let the cell uptake the DNA, and then washed away the cell thoroughly so that no more of that DNA is attached to the cell. So once it has taken in the DNA, they are now watching this endocytosed piece of DNA through or over time. So 5 minutes, 1 hour, 2 hour, and they see clear persistence over time in structures which have very low pH. So you see these structures in which it is now present versus 5 minutes or 1 hour. Now at 2 hours, you see it's completely present in structures which have very low pH. So this is kind of a pilot experiment just to show you that this is possible. It is possible to observe pH changes inside the cell using this particular DNA construct. Now you can design various things. Suppose you want to have a certain protein and you would like to see where is that protein going and is the local environment of the protein becoming more acidic or remaining close to neutral. You simply attach this DNA molecule with that protein of interest and you follow the fret as this protein is endocytosed in the cell and moving around in the cell. So there are plenty of things that can be done using this pH indicator molecule. And the pH indicator molecule, the entire construct, again, I have to emphasize, depends on the base pairing between these 3 oligonucleotides. And this single-stranded piece of DNA is designed to form non-Watson-Crick pairing here. And this is dependent upon the pH of the environment. So if the pH is low, then this pairing is preferred. And if you add fret pairs or a donor-acceptor pair to the end of these oligonucleotides, then once at low pH it assumes the structure, then the donor and acceptor molecules will be at close proximity and you will see high fret. So with that, I will stop my lecture. If there are any questions, we can take it. No questions?
Ma'am, can we use these DNA walkers to deliver drugs like in the cells? Someone has done.
I do not know if somebody has done that, but this is a cool study, right? You can at least in silico, you can think of it as being able to deliver movement in a particular direction or deliver movement with a certain velocity, because you can create these specific movements at specific intervals. You can also control the velocity by controlling the flow rates of the first attachment, second attachment, detachment, etc. So that can be achieved in vitro. In cells, I don't think anybody has tried, but I'm not sure. It's in cells, I don't think anybody has tried.
Yeah.
In cells, it will be also quite difficult. It's not impossible. You can think of it as, say, in a dish, you have these, and then suppose you have placed a cell at the very end and you attach the dye, or let's forget drug, let's think of it as a dye, and you see that the DNA then goes and attaches to one part that is very close to the cell. Yeah, that maybe it's possible, but directly inside the cell will be very hard to design.
Yes, ma'am. Yes.
Any other questions? All right, then I wish you all the best for your majors. Your majors will be on 10th, and I will send you a mail shortly, and I will also upload it on Moodle, the instructions for your majors. As I explained, the major will be open book, open text, open source, open Google, everything. Doesn't matter as long as you're not copying from each other. So please do not copy. If you're copying from each other, I will give both the persons a straight 0. The second thing is that please take your time to write the exam, because I literally have to read every sentence. So last time I found it a little difficult to follow the handwriting of a few people. So please take your time to write it. You will have 3 hours to write the exam, although the exam itself would be, the question paper would be such that it can be done in 1 hour. And Friday would be a discussion session, which means that if you have any doubts, you can clear them. I will hold the discussion session, but I would request you all to submit your questions beforehand. This would allow me to also if there are like 4 or 5 questions that have the same kind of answers, or if there is something that everybody is confused, maybe I can re-examine that entire topic once more. So if you have questions, please send them once more to me, and I will take them up on Friday. So please send them to me, if possible, by Thursday morning, so that it can be taken up on Friday. All right. Thank you, students. Thank you so much.
Thank you, ma'am.
Thank you.
Thank you, ma'am.